Welcome, everybody, and thank you for joining our webinar. Today, we have a very special guest. My good friend and one of my favorite Bayesians, Eric Ma, is with us. I personally known Eric for many years now as a fellow PyMC co-developer, and these days we're also working together at PyMC Labs, a Bayesian consultancy. And I'm particularly excited about chatting with Eric because at Labs, one of the things we noticed is that now that we're really getting a lot of experience with working with Bayesian methods in industry, is seeing what type of industries actually are really most interested and where Bayes seems to have the biggest impact. And one of those definitely biotech. And that's why I was like, well, the perfect person to talk to about that is Eric, who has all that experience through his work at Moderna and Novartis. So that's what we'll do today, is just basically get his insights into the why Bayesian is so popular in biotech and what makes it great. So welcome, Eric. Really happy to have you and very eager for your introduction to our listeners.
Yeah, likewise. Hey everyone, I'm Eric. I currently work at Moderna. I'm also helping out with PyMC Labs and work with Thomas on some of the biotech consulting projects that PyMC Labs engages in. I personally have really enjoyed contributing to PyMC. I remember my very first pull request, which was fixing a multinomial sampling bug on CPU versus GPU. It took me about 4 days to debug fully, but it was worthwhile. Really got me into the internal
first contribution.
PyMC. And it's been a great learning journey. Personally, I did my grad school at the MIT Biological Engineering Department, where I did my thesis work on influenza. Did not have to use Bayesian methods there, but I did have a lot of time in grad school to learn stuff. So that was like a very, I think for those who are thinking about grad school, by the way, that's possibly one of the advantages that you can have doing a PhD or doctoral training. You actually have quite a bit of time and self-direction to pick up new skills, Bayesian methods included. And it's as we'll know, like as we're seeing, it's actually a very valuable skill to have, as far as I've seen as well. Then I went on to Novartis. Now I work primarily at Moderna, also helping with labs. My professional mission has always been to make science run at the speed of thought. And it's my conviction that proper modeling methods, which really just means Bayesian methods, are the only way that we're going to get to that, make that a reality.
Great introduction, thank you. So let's dive right in and maybe start with an overview of which examples you've come across in biotech where Bayesian modeling has been particularly useful.
Absolutely. So I think we can maybe start off with the major categories, and then I'm happy to delve deeper into any of them. For those who are familiar with biotech, the biotech industry, you'll know that there are a lot of assays that are run. And assays are nothing more than just experiments that we do on molecules to test them for certain properties. That data turns out to be very expensive to generate, and it turns out to be you don't get large N data, you get very small N data. And that is the first place where Bayesian methods can be really helpful. And in particular, I'm going to reference this over and over. I'm a proponent of the use of Bayesian estimation methods. Bayesian estimation actually started in the psychology world with John Krushke, with his best paper, Bayesian estimation supersedes the t-test. And if you just think about Bayesian methods in its logical form, you'll actually realize it extends to every single kind of estimation problem that you can get. Every type of experiment that you do in the lab can be analyzed using Bayesian estimation methods. So that's the first one. The second place where I see Bayesian methods, and maybe we'll need to broaden the lens a little bit, probabilistic methods, is actually in like building models of analytical chemistry data. So when you're doing analytical measurements, you're going to get typically curve over time, or you're going to get some form of curve that looks like probability density function. And you're going to need to, and there's a formulation of analytical chemistry problems where what you're trying to do is you're trying to like decompose this curve into its components. You're effectively doing clustering, but you're given the empirical PDF, not draws from a mixture model. But that is another place where if you cast that curve as looking like a mixture model, you can actually, a probabilistic mixture model, then that's actually a really powerful way to decompose your analytical chemistry data. It appears Thomas might have dropped off the call, but I'll continue for the sake of the audience. The third place that I think we've seen a lot of success is actually in process modeling. So that is to say, you have a laboratory process, or more generally speaking, a business process of some kind. That business process can be a research process, it can be a clinical trial process, for example, and you want to be able to generate a model of the system that you're trying to analyze, such that you get back parameter estimates of interest on which you can intervene. So essentially, it's a causal model, right? Like you're building a causal model of the system. You're not using simple linear equations. You're actually explicitly modeling every step of the process, and that inherently is causal. And once you have that model in hand, you can actually go on and do a thing here where you can do these what-if scenarios. What if I increased, for example, the enrollment rate of patients into a clinical trial? What if I decreased or increased the screening success rate of patients through a clinical trial? Or what if I increased in a portfolio of projects? What if I did some things like increased financing on a project to increase the success rate in this department? That is a third place, process modeling, where Bayesian methods where I've seen the use of Bayesian methods, and I have this, and I've seen their success actually in being able to say not only what the estimates would be for key parameters, but actually taking it all the way through to the end. It's like, what is the impact on the business, right? There are certain processes for which you absolutely need to know and quantify what the right tail of the distribution might be, right? Because that concerns overproduction of stuff that you want to overproduce. You don't want to underproduce supplies for a clinical trial, for example. But then on the other hand, when it comes to like staffing and stuff, you don't want to overspend. So you need to know the left tail of your forecast distribution, and propagating that uncertainty through all the way turns out to be a really, really important step as well. So those are the three: Bayesian estimation for for assay data, 2 probabilistic models of analytical chemistry data, and 3 process modeling, where you're able to leverage the uncertainty at the end of the entire process, where you have the causal model of your system written out in equations, and you can intervene on there right at the end and go like, what if I did this? What if I did that onto the system? So those are like 3 places, and where the uncertainty is also useful. Those are the 3 places that I've seen like really powerful usage.
And I'm curious you already alluded to this a little bit, but if you can extend a bit to why in particular Bayesian methods are very powerful.
Oh yeah, absolutely. There's this myth that pharma and biotech have large amounts of data. We have large amounts of data when viewed from a certain angle, but if you think about like the experimental nature of biotech, where especially for me, I work in the research space, so we're asking all these causal questions of our molecules. Does this molecule cause a decrease in certain output in a cell of proteins in a cell, or does this molecule instead change gene expression and stuff? You actually don't get to collect a lot of data per molecule, because budgets are limited, and budgets are limited primarily. So like the amount of money and time that you can spend on like characterizing a single molecule is very, very little. And we have this concept of a funnel where you have to like progressively narrow down the things that you take forward, the molecule candidates that you take forward, because the experiments that you need that will tell you what's going to happen further and further down, like closer to the clinic, get more and more expensive. You start with experiments in a dish, then you progress to mice, and that's like 1,000-fold cost increase. Then you go from mice and you go to primates, and that's another 1,000-fold cost increase. Then you go from primates to humans, and that's another 1,000-fold cost increase. So you're going like, it's a billion-fold increase basically when you go from like molecule in a petri dish to molecule in a human. So you actually don't have the full spectrum of data that you might want to have. And so here's where, if you don't want to be fooled by randomness, I've been clearly influenced by a book there. If you don't want to be fooled by randomness, you really want to have methods that properly quantify the uncertainty that you have in your estimates early on and de-risk your experiments early on in the research phase before you go to clinical development. And if you don't do Bayesian methods and instead counterfactually you did, say, frequentist t-tests or ANOVAs and whatever without properly considering the data generating process involved, you're prone to, first off, errors in modeling, and secondly, errors where you're fooled by randomness. And so I think that's one reason why Bayes in biotech has been gaining steadily more and more attention. It's that people are waking up to the fact that t-tests everywhere and ANOVAs everywhere aren't just going to cut it anymore for the kind of data that we're getting that is not necessarily going to be Gaussian distributed, right? Imagine trying to do the t-test on Poisson distributed data. Like, well, your prerogative if you want to do it, but you're going to do something wrong, horribly wrong, from a modeling perspective, and you're going to be fooled by randomness at the end. So I think that's like the big thing here is that two reasons then, right? Like we have small data. Like I'm just going to call it right out. We have small data in biotech. You really can't gather a lot of data, and I'm happy to talk about that later. And secondly, the canned statistical tests really don't cut it anymore. People are starting to recognize that there's a diversity of data that's generated that is not necessarily continuous, and applying canned statistics on that data just doesn't cut it anymore. So I think those are the main reasons why I think Bayesian methods are gaining more and more popularity nowadays.
Yeah, that definitely matches my experience and with talking to clients where, individual data points can cost tens of thousands of dollars. So getting really the most out of it and really leveraging all that understanding you have from, hundreds of years of science, right? We really understand a lot of these things at a very deep level already, and it would be somewhat wasteful then to throw that away. And with Bayesian, we can build a model that incorporates all that knowledge that we have about the process, and then really just like have those few data points fill in the rest.
Yeah, absolutely.
So
and I think you raised a really great point right there, because there's this concept of like a digital twin that's gaining popularity in the lab, where you're able to replicate digitally all laboratory processes. There's another layer of digital twin here, right, where you've got your mental model of the biological system. And if you translate that directly into mathematical equations, now your mathematical equations are a digital twin, exact digital twin, sorry, a highly precise, that's the best definition I should give here, a highly precise digital twin of your biological system. And we all know, like from the pitfalls of machine learning, which I'm a fan of, but I'm not a proponent of everywhere. The pitfalls of machine learning are that if you don't get the causal model right, then your predictive model is always has this risk of being wrong. But if you get the causal model right, your predictive model's very, very unlikely to go wrong, right? So having that digital twin of a model, so like if your system is inherently Markovian, you don't go in and just like place Gaussians all over the place. You write the Markovian model equations out, and you model the transitions, model the emissions correctly, and now you're able to maintain this digital twin and intervene on the system digitally before you do any laboratory experiments. And that's an incredibly powerful thing to have, to be able to simulate what should go on before you go and make any decisions about what do you do with your molecules.
Yeah. And also really powerful. I always find that once you have it, it becomes so much easier not only to reason but also communicate
Yes.
the assumptions in your analysis to other people, right? You just like show them like, well, this is how I think everything is connected, and maybe they disagree, and then you can have a very productive discussion of how they think it should be, and you can try both of those and
Yeah.
maybe compare results. So yeah, it
Interpretability.
seems like a very way. Yeah,
Yeah.
exactly. Actually, I just because you embedded things in a causal language, I wanted to just like quickly make a detour there
Mhm.
because I think I mean, it's definitely something that has been of interest to me, but I think there's like just a lot of confusion out there where it's like, well, there's causal modeling and then there's Bayesian modeling. Are those different? Are those the same? Are they compatible? Are they incompatible? And like there's all kinds of answers you can
Yeah.
find. So I was just curious what your
Yeah.
opinion on that
Yeah.
is.
I don't tend, I'm not trained in causal inference, but I have read some literature, and what I think is correct is that causal inference methods have usually been concerned with inferring the causal structure and therefore the equations that you need to model a system from observational data. That's one way of looking at like causal inference, causal like modeling. That is, I've just got observed data and I have no prior structural information about the data generating process. And I want to infer what the structure of the data generating process might be. The simplest of causal methods, which are the only ones that I've studied before, are linear models everywhere. And it's not powerful enough for the kind of like the simplest causal inference methods are not going to be powerful enough for what we are thinking of doing in like biotech. So there's a second angle to approach this like causal modeling thing, which is you think like a physicist instead, right? Where you're thinking like a physicist and you're writing down physics equations, right? And or instead of physics equations, you're writing like dynamical equations, or you're writing Markovian equations from first principles, your prior knowledge encoded in your head in the structure of the equations, right? And that is a second way of approaching causal modeling, which is like, forget about inferring the causal structure. I believe there's a causal structure and there's a structure of equations that takes us from parameters to output. And that is a prior assumption we make when we're doing these modeling things. Like the simplest is a first order decay for exponential decay curve, right? For whatever thing that might decay. Like you wouldn't go in and you would write a linear model in log space, for example, but you wouldn't go in and try to discover that structure. It's like a primary belief that you have walking into the problem of like measuring the decay rate of a molecule in a body, right? So you would in a petri dish. If you go to the body, then you'd have a more complex set of equations that you need to consider: liver clearance, transport through different organs, sequestration inside fat cells, and you'd write instead dynamical equations, differential equations to model that. And so that's a second angle, right? The first angle is more social science oriented, which is like, I have absolutely no clue what the causal structure is. And the second is more physical sciences oriented, which is I have very strong beliefs about what the causal structure is. Now, that never touched on Bayes, right? So where is Bayes in all of this? Well, it turns out once you have the equations, you have parameters to estimate. If you believe your parameters are like, if you're absolutely 100% confident in a point estimate of your parameters, you have no need for Bayes. But can we ever be 100% confident in our estimate? I don't think so, right? We have uncertainty. So as soon as you need to model the uncertainty in your parameters, you introduce Bayes. You introduce a likelihood function for the observed data that you have, and then you like walk backwards and ask what kind of distribution would my parameters be drawn from, and that turns out to be a much more clarifying framework for me to think about causal methods and its relation to Bayesian inference. Causal inference at its core is causal structure discovery. There are other parts of causal inference like estimating treatment effects and whatever from observational data. But as soon as you've got equations to describe your system, to take it Bayesian, just say that we don't precisely know my parameter values, and then start figuring out how to quantify your parameter values. And it becomes really much simpler for me at least, my little brain, to think about the relationship between these fields in that framework instead.
Yeah, I really like that answer. And a lot of times they are treated as alternatives, but really I think they're like different layers of the cake where, you can embed this in a Bayesian framework and get all the goodies of Bayesian modeling. I guess there's at least one other thing that people often care about in causal modeling, which is the do operator, where you
Intervene.
Imagine you would have you intervene, exactly. And just mention it here. So Ben Vincent is currently working on a really exciting blog post that highlights PyMC functionality that allows you to do the do operator and how to do causal modeling with PyMC. So that will be on the PyMC Labs blog post. I don't know when, but probably in 1 or 2 weeks. So definitely check that out if you're interested in it.
Yeah, I've talked with Ben a few times about causal modeling and that sort of thing, and it's like really, really cool. Ben has really like run with it. It's so cool to see the body of work that he's developed, the tooling that he's developed around this.
Yeah. Exactly. So I guess that answers one of the questions here. Is there a causal model example in PyMC docs by Kat? So thank you for asking. So there is actually some examples already, but they are more quasi-experiment ones. But the more proper is coming up. So yeah, stay tuned on the PyMC Labs blog post and the social media where we'll post this. So I want to take things back a bit now to your work at Moderna and what you term research data science. And I'm just curious, basically, what do you think of that term and what that looks like at Moderna and Novartis?
Definitely. So all my professional career, I've only worked in research type organizations. I've not worked at places that say do advertisements, marketing, or optimization of retail, etc. So I've only ever done it like with in the research field, and that partly stems from that my brain is a nerd and wants to deal with molecules and proteins and not deal with like people data, for example. But the short answer of like research data science is, in my head, data science that accelerates science to the speed of thought. So my overarching mission, professional mission, as I mentioned at the beginning, has always been, can we make molecule discovery science run at the speed of thought? If we can do that, we can make medicines for patients more cheaply at lower cost, for a greater range of diseases the impact is just like better if we can make science be less laborious, more automated, more principled, all at the same time, not having to sacrifice any of the others. So maybe what would really help here is if I gave just a very brief background about like what is the framework by which we think about drug discovery problems, right? There's a therapeutic holy trinity. You start with an indication, which is nothing more than a disease. For that indication, that disease is always a dysregulation of the body, right? From its normal regulated state. So for that disease, there's always a hypothesized target or targets for which those specific proteins in the body or that tissue or that like organ system is dysregulated. And you really want to go as close to the gene or the mRNA, the RNA, or the protein you want to get as close to that as possible. So for a disease, you want to find a target that is dysfunctional in the body. And then what you want to do afterwards is find a molecule that corrects that dysfunctional component so that you bring the body back into a properly regulated state. A lot of drug discovery, as I've seen it at Novartis and as I've heard from friends at like Pfizer and Merck and other Takeda and other companies that are in the local biotech area in Cambridge, is primarily concerned with research is primarily concerned with target identification and molecule identification, right? So like target identification is which target do we need to hit to correct, do we need to hit in order to correct the dysfunctional state? And then the molecule is what molecule do we add that will hit the target that will correct the dysfunctional state. Molecule work is the place where I've placed the most amount of my headspace, which is to say I've got screening data and I need to do two things. Screening data, by the way, is I measure up to 15,000 molecules in an early discovery assay, in an early discovery experiment, up to 15,000, 20,000, 30,000 molecules through robotic automation and such. And I measure them by systematically applying these molecules to like model systems in a petri dish, very small petri dish, 96 well plates. We're thinking 96 to 384 well plates, and you just got stacks of these plates. And you do this to, you do this exercise where you're like screening all these molecules to figure out from all of this assay data, which molecule can I apply to the system that will correct, potentially in a human later downstream, correct the dysfunctional state. We're still, keep in mind, we're still talking about like cellular models, right? And in that regime, you're lucky if you have two measurements of the same molecule. You're even luckier if you have three measurements of that one molecule, so replicate measurements in a system, right? Because the logistics of some of these assays, if you're doing like an inhibition curve assay, you're not just applying the molecule once at one concentration and measuring at one time point. You're taking that molecule, diluting it 6 different times and measuring at a certain time point that you've had to figure out beforehand in order to understand when to capture the data. So it's like very, very complicated, right? And you actually have from like 15 to 30 measurements, you have this step in the middle where you have to estimate the core parameter of interest, which is, say, the inhibitory concentration 50, IC50 of that molecule. So 18 data points potentially over 9 time points gives you duplicate curves. If it's 18 time points over 6 time points, that gives you triplicate, but you're lucky if you can get anything close to that quality of data, right? And that's why it's so important to quantify the uncertainty in your IC50 estimate as it is measured throughout the entire system, right? And so that is like one example of like research data science in action where the Bayesian methods have to be applied early on, right? Plus, don't forget, this is not just one molecule. This is not just one molecule at one time point. This is 18 molecules for one time point. 18 measurements of one molecule. Multiply that by 15,000. And now you have like very sparse data covering lots of molecules. Each molecule is your sample, and you need to go in and estimate what is the IC50 for each of them. If you're not careful, you're going to be fooled by randomness. And therein, like bringing things like hierarchical models and the likes really helps with this estimation task. It's difficult to write those models, by the way, but like once you get your head around the math there, you're able to write the vectorized version of those models. It's actually not difficult with PyMC to just fit a model, and I've done that before. But yeah, that is like an example. That is like a view on what research data science looks like, right? Then there's also this problem of like, okay, what do I reprioritize for further measurement, right? If you don't have uncertainty in your estimates of, say, IC50, you might, by lucky chance, get a molecule that has like has a very low IC50, which is sometimes desirable, right? And if you're going for low IC50 and by lucky chance you got your low IC50 thing without doing Bayesian hierarchical modeling for regularization, you're going to be fooled by that estimate, right? And that's not a great place to be in when like the cost of failure is extremely high downstream, and it just increases over time. So getting your Bayesian estimates in early and putting applying proper regularization early on your estimates is of value. It's just difficult sometimes to quantify the value if you don't have external research portfolio information, but one can make that case already just through pure logic that if you don't get your estimates right early on, and we're fooled by randomness more than once early on, we're going to pay a very large price, a billion times larger price down the road when we go into humans.
Yeah, I like how you connected this with the beginning, right? Because these things are extremely consequential if you go down the wrong path, and it takes you I don't know how many steps until you finally at the very end in the human trial, clinical trial 3 maybe, you find out that it doesn't work. Then that is easily $1 billion down the drain. Also what I thought was really interesting is you said little data, but and this is, I think, really an often overlooked point, is that certainly you don't have a lot of data points, as you mentioned. So you don't have long data, but you still have a lot of them, right? So you have wide data. And that is probably the case where especially frequentist methods have the most difficulty, because you just get into type 1 errors. And where, as you mentioned, regularization becomes so absolutely critical. And hierarchical models are just an extremely elegant way of doing that. Of course, you can do something similar in a frequentist framework, but it's just oftentimes a bit harder to express.
Yeah, it actually feels like in a frequentist framework, you're playing the game of Twister. You're twisting yourself left and right on your mat to get into the right place. Whereas if you're well trained in logic, the Bayesian framework, just makes sense. You logically add in a hierarchical, you may have started with a non-hierarchical model, but then as soon as you realize you're going to very wide data, it's so natural to just bring in, oh yeah, that parameter, it's not independent per molecule. We believe it's drawn from the same distribution for this reason. It's all logic. It's like Jaynesian logic. There's no weird limit of how many experiments, infinite number of experiments thing that we have to deal with to justify doing that in maybe a frequentist framework. But it's just so much more logical, and it actually takes a little bit of try it if you've not tried it for yourself. Try it and work through the logic yourself, and then you'll come to a realization that there's no other framework that just fits the human mind. It's what do you call that? It's a tool for the mind, right? Yeah,
definitely. And you mentioned that you are using PyMC there, which is cool. Actually, I wasn't totally aware that it's being used at Moderna for drug discovery. And I'm definitely a fan of Moderna ever since the mRNA vaccines came out. So that is really amazing to hear. How has your experience been? How are your colleagues taking on to it? Are they? And has it been useful? And
absolutely. So I was originally drawn to PyMC because of the wealth of tutorials that were available. Other probabilistic programming languages that existed in 2015 just weren't as accessible. And I think people underestimate the power of a very well-designed language, right? If you think about how PyMC's syntax looks like, it's very natural to translate between mathematical equations if you're skilled at writing them, and PyMC equations. And even if you're not skilled at writing math equations, which that's me, right? I'm very algebra blind. I will make all sorts of sloppy algebra mistakes. It's the cause of why I've struggled to get an A+ and have always Asian failed my math classes. The cool thing about PyMC syntax is that if you think more like a computer program, it's also very logical. You can trace your flow of parameters through the model once you've got the model structure all written out. And that context manager is genius. Man, it's just genius. I really appreciate. I've only, like, believe it or not, 10 years of programming in Python. I only this year learned how to implement a context manager, and once I learned it, I really appreciated how elegant the PyMC context manager syntax is. And so at where I work, Moderna, which is my primary day job, I've coached my teammates on how to use PyMC, and I've insisted that we use PyMC because, firstly, I'm on the dev team of PyMC, so I'll be able to really know and pull in the latest information and have firsthand information about, like, latest developments of PyMC, and we can start incorporating those. So, for example, find priors was a thing that, like, I had been privy to discussions about, and prior to, sorry, bad pen, but prior to find priors, the way that you find priors was kind of weird, right? You had to, like, draw it out and guesstimate. But now find priors, the function, we use it to find priors in a way that we just declare in a very logical fashion. My prior should have some shape, this distribution, and fall between these two values as the most likely prior belief, right? And that just turns out to be really, really powerful, right? And having firsthand access to new developments in PyMC is also, like, really, really cool. And if you pitch for PyMC Labs, if you work with PyMC Labs, by the way, you will get that firsthand information as well, because the large proportion of the PyMC core dev team actually works with PyMC Labs in some capacity. So it's like a lot of advancements, also practically useful advancements, are being ported into the PyMC library or the PyMC experimental libraries, and things like the do operator for causal methods, like in applied settings relevant to business problems, will be in the library for free, right? So it's like a really, really cool operating model there. Thomas, I don't know if you wanted to, like, continue the pitch, or if you want me to continue on.
Just one thing which is dear to my heart is that this has really been amazing to see, that it was really one of the big motivations and visions for starting PyMC Labs is making PyMC better for that particular business use case. And that has really panned out, where through working with clients, we discover some shortcomings in the library, things that could be improved, and then we have the resources now to do that and really just make it better. And in the last a couple of years, like the library has progressed immensely, and yeah, that's just very exciting.
Yeah.
So
yeah, definitely. And so at work, I do try to teach my colleagues about Bayesian modeling, and more generally, my approach has always started with, like, you got to work on a project. Like, if you don't work on a project, it's not going to work. So my teammates, I really try to find projects for them where there's a high likelihood that they will be able to apply probabilistic methods onto the problem in whatever form, right? And then secondarily, less effective, but still nonetheless valuable, is doing workshops, internal workshops of some kind. My bandwidth is limited, so sometimes it's not possible to do it at the frequency that I would like to have. But nonetheless, I do know that some people learn better in a formal classroom setting where there's, like, structured exercises and that sort of thing. And so I try to conduct those. I know Labs does workshops for clients as well. So, like, if your teammates or team members are more comfortable with that style of learning, that's another great way to pick it up. And those are the ways that I try to encourage the use of PyMC. There is some debate about, like, whether we can join probabilistic models with other machine learning systems. One example being, at Moderna, we use PyTorch. I've wanted to use JAX, but many machine learning models were already written in PyTorch, and so be it. We use PyTorch everywhere there. And so when it came to the decision on whether to use Pyro or to use PyMC, there was a little bit of debate, and some people actually favored Pyro over PyMC for the reason that it's all PyTorch underneath the hood. Now, I'm a NumPy kind of person, and I don't like the PyTorch API as much as I do like things that stick to the NumPy API, because if I'm a believer in like, if your language follows language idioms, then like it will make adoption so much easier. And I don't understand why the PyTorch world did not just go with the NumPy API. But PyMC and the previous iterations of the underlying tensor library, so PyTensor, Asara, and Theano, all had Theano did not have a strict policy of following the NumPy API, but Asara did, and now PyTensor as well, right? And that's like, makes me happy, because NumPy is the lingua franca of the PyData stack, not PyTorch. PyTorch is and so if you stick with the NumPy API, then you're actually going to do things a lot more accessibly, right? So I think there's maybe a little bit of work that needs to be done on PyTorch's side to adopt the NumPy protocol. I think that's NEP 12, if I remember correctly, NumPy array protocol. And at the same time, I think if they do move in that direction, there's an opportunity for, say, PyTensor, which already wraps JAX, Numba, and some C++ libraries, to also hook right up into the PyTorch autodiff capabilities. And that might be a great direction where we'd be able to like really join up probabilistic models. If you had an easy way to wrap a PyTorch model and have its gradients flow through as part of your like full-on predictive model that includes the estimation portion and the, you say, molecule structure or protein sequence to assay result prediction. Oh, that would be really, really powerful, right? And I can see that might be like a great way of unifying the two halves of my day-to-day work, which is Bayesian estimation and then predictive modeling. Predictive modeling right now is entirely in PyTorch. Bayesian estimation is entirely in PyMC. And I'd really like to see the two of them joined up in a little bit more unified of an interface.
Yeah. And Max Kocherov actually had the idea of adding a PyTorch backend to PyTensor and to PyMC, which I think makes a lot of sense. I mean, with PyTensor now, one of the strengths of it is that we're not just committed to only one backend. So it used to be just C.
Yeah.
But then Brendan Willett and some other folks have added like a JAX backend and a Numba backend. And these have really like panned out extremely well in terms of scalability and being able to run models on the GPU, and also incorporating now JAX libraries. So if you run with the JAX backend, you can incorporate JAX libraries. So if we had that for PyTorch, which wouldn't be overly difficult to add, you could definitely like hook up and wrap your PyTorch models to be included in the PyMC model, and everything just like runs in PyTorch on whatever compute execution engine you want, which would be a really nice world. And this is actually something which I also think is absolutely fascinating. I haven't really seen it being used as much as what I started calling hybrid models, where you have, because there's really no thing where like either like machine learning is better or Bayesian modeling is better, like they're just different for different type of problems. But certainly they can be complementary, right? So at the very simplest level, I guess you could have first, let's say you have the Bayesian model, which might be linear, right, and takes out all the information or models all the information that you already know about the problem, right? Like all these good things with the digital twin we talked about, and then basically the outputs of that. There might still be something left, and if you just want to squeeze out the predictiveness, you have a model for which you have no real interpretability, but nonetheless you want just to maximize predictiveness, then just combine those two. So that seems to be something that you also thought about.
Yes, absolutely. And like that is ultimately the end goal of like research data science, especially when as applied to molecule discovery, protein sequence space, molecule structure space, even for a given protein, the mRNA codon space. Those are all way too many possibilities, combinatorially large spaces that you would need to explore. The value proposition of machine learning methods, in this case here, is to be able to shortcut your exploration. So you don't have to go through a random search through chemical space or protein sequence space or mRNA codon space. You can actually focus more on a bit more exploitation of local spaces that might get you to a place that is good enough, right? And that is a great like application of the machine learning tooling, and I'm like all for it. I just want it also, I think personally, I also just wanted to see that our predictive models can be written in a way that's more unified with the estimation models as well. Like if we can do that, that would be, you would have a very, very strong causal model, because your protein sequence is causal for whatever output that you have, or your molecule is causal for whatever output you have. That's why you're able to design an experiment where you intervene on the molecule and you switch the molecule out, right? And get a different assay result, right? So like, if we can do that, that would be amazing. That would absolutely be amazing.
Yeah. I guess we'll see whether the research catches up, because I haven't really seen that being discussed a lot.
Yeah.
So all of that is a lot of premise.
Yeah.
Is there anything where you think we need to work harder, or like anything that you think is holding Bayesian modeling back for even wider adoption? Like, is it the tooling? Is it, I mean, for example, you already mentioned the training. Like, I can't imagine that it is easy to find people who like know a lot about biotech, and then also like trained statisticians with Bayesian modeling background, and probably also some good software engineering mixed in. Right. Like that's very hard to find.
It is. I think it's people are the problem. That's like, I don't mean to put it in like a derogatory fashion.
Period.
Like, finding people is a huge problem. In my experience, finding people who are skilled in Bayes is already a rare thing. Then if you do the set intersection with people who are knowledgeable of and have graduate level biochemistry, biology, or chemistry, or like genetics, that's an even smaller set of people. And then here at Moderna, I'm very thankful. Like, we treat all of our data projects like software, right? And there is a software product that comes out at the end, whether it's a command line tool or a Python library. Like, those are our work products. And finding someone who also comes in equipped with that skill set, just as you mentioned, is like a third set intersection to do. So you can imagine the conditional probabilities get really small there, right? What I've tried here is to prioritize finding people who have at least gone through some graduate level biology or chemistry, because on that front, I know I cannot teach the breadth of biology and chemistry knowledge that is needed to deal with that we deal with in our organization. There are too many. There's epigenetics, analytical chemistry, organic chemistry. There's cellular biology. There RNA biology. I can't train on that front. On the other hand, if it comes to a smaller core set of like software development skills, that's a little bit easier. You don't have to, when interviewing, for example, I don't have to go in and make sure they're like writing code with recursion and that like context managers. Like, that's not necessary. They just need to like or have a bunch of functions that are organized well, and I can teach them the rest, right? And now where it comes tricky is probabilistic modeling. It's not taught enough, I think, in our formal university curriculum. I remember when I was an undergrad, stats 200 was taught essentially as, here's the t-test. And I was like, I scored an 88% on that class because of sloppy algebra. That's another math class that I Asian failed. But I left that class utterly confused about statistics, right? And not knowing what the difference was between the standard error of the mean and the standard deviation and whatever. It's like very, very confusing because you had all these things to memorize. We don't teach data analysis in as logic in our undergrad curriculum. And that's setting us up for a pipeline constriction of people who then move on to like higher level, upper level undergraduate statistics where they may introduce Bayes' theorem, and then setting us up for an even more bigger pipeline constriction where like only a subset of those will go on to graduate training in some comp bio or statistics program, where they're trained in Bayesian methods, right? So I think there's an education problem, and that's what I mean by a people problem here. So the pipeline of people who are familiar with the methods through a formal training is like very, very limited. And then there are other people who, like myself, have had to pick it up on the side. So I picked it up during grad school, and then like more nights studying on my own, working through tutorials, making my own examples just to make this like Bayesian thing make sense. That takes time, and it takes a certain type of motivation, desire to learn, and not most people will have their motivation to learn eliminated once they're in the workplace, right? And so it's like it's a very tough problem, and I think it's the people problem that is going to be the primary problem. And I think where we can work on, as practitioners of Bayes, is to go in and start training people and ignore the voices that go like, oh, couldn't you just do this with some fancy, then like memorized fancy name test, or like some memorized fancy name method. Forget about that. Like teach the logic, teach probability as logic, teach data analysis through logic, in our day-to-day work. Try to bring into, through workshops, through PyCon, SciPy tutorials, which are recorded and free, which is how I learned, right? Like that's how I learned that stuff, PyMC and Bayesian modeling. I think we need to put in a lot of effort on the education side. That's my biggest, that's what I see as the biggest hurdle. And then having more people talk about the power of Bayes in their work is also a great thing, like today.
Yeah, I definitely agree. And this is actually a good segue to a new thing that we're trying is if you are interested in this, but you're like, well, this sounds like a lot. But I'm not quite sure where to start, or is this really applicable to my problem? Then we'd love to just help you with that, basically. So for everyone here, if you're unsure, there's a thing where like we go for 30 minutes and we look at your problem in depth. And it says marketing analytics pipeline, this is wrong. Your biotech analytics pipeline and how Bayesian models can help and what type of Bayesian model you might need. So there's been a lot that we discussed today. And really come up with a roadmap. And the underlying theme here is really the dissemination of knowledge of these tools, right? So obviously, Eric and I think that this is very powerful, and you might have the thought, but then how do you do it, and also how do you find the people, right? So that's basically what PyMC Labs is about. Is we have the people specifically in biotech, like Eric and Adrian and many others in the team have extensive knowledge in biotech, and these are some of our favorite clients to work with. Just because, well, it's a super interesting domain. It's super. The Bayes often makes a really big difference, which of course has been the topic of today. And yeah, so if that is of interest, just check out calendly.com twiki Bayes. You can book a call and then we'll just chat. So yeah, really happy to to take that and to get to know anyone here more closely. So that is that will be really fun. Then how do I stop? Okay. And then I guess we can go into some Q&A. So there's one question by Danny. Do you have examples of Bayesian models on RDF data? RDF data for biological designs, for example. So
RDF data.
Yeah. So yeah, RDF data.
Let me see if I can. Let me take a look at that question. Actually,
what is RDF?
Yeah. I am not sure what RDF data actually is. So let me think here.
Okay. Well, maybe Danny, you can specify.
Oh, yeah, RDF data for biological designs. I think Danny, if you could like maybe elaborate in chat a little bit what you're thinking of. It may be stuck somewhere in the depth of my head. I've seen RDF data before, but I think I'm blanking on it right now.
RDF is a form of storing data in graph structure.
Okay.
SBOL.
Bayesian models on on RDF data. So I personally don't have any experience trying to build Bayesian models around RDF data. What I'm probably more inclined to think of here is that the way that you store the data is less important than the structure of the laboratory experiment that you've got on hand, right? So like, if you're, let's say, for example, you're in the synthetic biology world and you want to estimate, you want to quantify the conversion rate of a protein that you've just engineered, like, and you've got 15 variants that you've tried, right? The way you store the data doesn't matter as much as whether you can shape the data, take it from whatever your underlying storage structure is, into a format that is long form, tidy data. If you're familiar with the R world, there is this concept of the R tidy data, right? Like Thomas, I think you may have heard of that before. And it's basically convert your data into a table that's long form, right? And if you can turn it, if you can write the code that goes from long form, from whatever graph structure into long form tabular data, you have a very good path for like fitting models around that, I think. Cool. And then I think there's a question from Scott. If you have a model like 50 and fitting parameters, is there any power in having, say, 6 different concentration triplet versus 18 concentration points, so long as all the concentration points are in a range that can help define the IC50? I think the answer is yes. That becomes an experiment design problem, right? Like when you're trying to do a concentration dose response, one of the first things you have to do is like do a range finding experiment before you actually do the characterization experiment. The range finding experiment is, what concentrations should I put my molecule in order to do what concentration should I be testing for the final actual dose response curve. And then if you get most of your data points, like let's say your curve was this shape and most of your data points were actually on the right hand side, you would not measure the IC50, right? So you need the range, you need the dose response curve in coarse grained form first, and then the most crucial part of the dose response curve is actually the slope downwards. So you don't really need a lot of data points on the tails, but you need more higher resolution data points, ideally in the middle portion of the curve. So there is value in that, and you can in doing triplicates. And if you do triplicates, it's even better, right? Like that is probably more ideal. But you also have to wrangle with another constraint, which is the concentration range that might be most relevant for one molecule might not be the exact same concentration range that's most relevant for another molecule. And yet the way, in order to do 15,000 molecules, you have to treat all the molecules through the same dilution series. And so that means sometimes sacrificing your resolution on the ideal dose response range and going to like larger, more coarse-grained measurements. So it's you know, Scott, I really like the question. It's also a very difficult one to answer in general, right? Like it's going to be very experiment specific, whether it depends on what molecule you're testing, whether you're doing this in high throughput or whether you're doing this in low throughput. It's all really going to depend a lot on that.
Great. Well, we're at time. So thank you so much, Eric. Thank you everyone for joining. I personally learned a lot. I hope the audience did too. So yeah, great as always chatting with you, Eric. And
Thanks a lot,
thanks
Thomas.
for coming on.
It's my pleasure.
See you.